Photosynthesis life defined. Photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that, through cellular respiration, can later be released to fuel the organism's activities. Some of this chemical energy is stored in carbohydrate molecules, such as sugars and starches, which are synthesized from carbon dioxide and water, hence the name photosynthesis, from the Greek phs, light, and synthesis, putting together. Most plants, algae, and cyanobacteria perform photosynthesis. Such organisms are called photoautotrophs. Photosynthesis is largely responsible for producing and maintaining the oxygen content of the Earth's atmosphere and supplies most of the energy necessary for life on Earth. Although photosynthesis is performed differently by different species, the process always begins when energy from light is absorbed by proteins called reaction centers that contain green chlorophyll and other colored pigments/chromophores. In plants, these proteins are held inside organelles called chloroplasts, which are most abundant in leaf cells, while in bacteria they are embedded in the plasma membrane. In these light-dependent reactions, some energy is used to strip electrons from suitable substances, such as water, producing oxygen gas. The hydrogen freed by the splitting of water is used in the creation of two further compounds that serve as short-term stores of energy, enabling its transfer to drive other reactions. These compounds are reduced nicotinamide adenine dinucleotide phosphate NEPH and adenosine triphosphate ATP, the energy currency of cells. In plants, algae, and cyanobacteria, sugars are synthesized by a subsequent sequence of light-independent reactions called the Calvin cycle. In the Calvin cycle, atmospheric carbon dioxide is incorporated into already existing organic carbon compounds, such as ribulose bisphosphate RuBP. Using the ATP and NEPH produced by the light-dependent reactions, the resulting compounds are then reduced and removed to form further carbohydrates, such as glucose. In other bacteria, different mechanisms such as the reverse Krebs cycle are used to achieve the same end. The first photosynthetic organisms probably evolved early in the evolutionary history of life and most likely used reducing agents such as hydrogen or hydrogen sulfide rather than water as sources of electrons. Cyanobacteria appeared later. The excess oxygen they produced contributed directly to the oxygenation of the Earth, which rendered the evolution of complex life possible. Today, the average rate of energy capture by photosynthesis globally is approximately 130 terawatts, which is about 8 times the current power consumption of human civilization. Photosynthetic organisms also convert around 115 billion tons 91,104 pg petagrams or billion metric tons of carbon into biomass per year. That plants receive some energy from light in addition to air, soil, and water was first discovered in 1779 by Jan in Genhouse. Photosynthesis is vital for climate processes as it captures carbon dioxide from the air and then binds carbon in plants and further in soils and harvested products. Cereals alone are estimated to bind 3,825 Tg teragrams or 3.825 Pg petagrams of carbon dioxide every year, i.e. 3.825 billion metric tons. Light-dependent reactions. In the light-dependent reactions, one molecule of the pigment chlorophyll absorbs one photon and loses one electron. This electron is taken up by a modified form of chlorophyll called pheophytin, which passes the electron to a quinone molecule, starting the flow of electrons down an electron transport chain that leads to the ultimate reduction of NAD to NADV. In addition, this creates a proton gradient energy gradient across the chloroplast membrane, which is used by ATP synthase in the synthesis of ATP. The chlorophyll molecule ultimately regains the electron it lost when a water molecule is split in a process called photolysis, which releases oxygen. The overall equation for the light-dependent reactions under the conditions of non-cyclic electron flow in green plants is not all wavelengths of light can support photosynthesis. The photosynthetic action spectrum depends on the type of accessory pigments present. For example, in green plants, the action spectrum resembles the absorption spectrum for chlorophylls and carotenoids with absorption peaks in violet blue and red light. In red algae, the action spectrum is blue-green light, which allows these algae to use the blue end of the spectrum to grow in the deeper waters that filter out the longer wavelengths red light used by above-ground green plants. The non-absorbed part of the light spectrum is what gives photosynthetic organisms their color, e.g., green plants, red algae, purple bacteria, and is the least effective for photosynthesis in the respective organisms. Z-scheme. In plants, light-dependent reactions occur in the thylakoid membranes of the chloroplasts where they drive the synthesis of ATP and NADH. The light-dependent reactions are of two forms, cyclic and non-cyclic. In the non-cyclic reaction, the photons are captured in the light-harvesting antenna complexes of photosystem Roman II by chlorophyll and other accessory pigments, see diagram at right. The absorption of a photon by the antenna complex loosens an electron by a process called photoinduced charge separation. The antenna system is at the core of the chlorophyll molecule of the photosystem Roman II reaction center. That loosened electron is taken up by the primary electron acceptor molecule, pheophytin. As the electrons are shuttled through an electron transport chain, the so-called Z-scheme shown in the diagram, a chemiosmotic potential is generated by pumping proton cations H+ across the membrane and into the thylakoid space. An ATP synthase enzyme uses that chemiosmotic potential to make ATP during photophosphorylation, whereas NADH is a product of the terminal redox reaction in the Z-scheme. The electron enters a chlorophyll molecule in photosystem I. There it is further excited by the light absorbed by that photosystem. The electron is then passed along a chain of electron acceptors to which it transfers some of its energy. The energy delivered to the electron acceptors is used to move hydrogen ions across the thylakoid membrane into the lumen. The electron is eventually used to reduce the coenzyme NAD with a H+ to NADH, which has functions in a light-independent reaction. At that point, the path of that electron ends. The cyclic reaction is similar to that of the non-cyclic, but differs in that it generates only ATP, and no reduced NADH is created. The cyclic reaction takes place only at photosystem I. Once the electron is displaced from the photosystem, the electron is passed down the electron acceptor molecules and returns to photosystem I from where it was emitted, hence the name cyclic reaction. Water photolysis. Linear electron transport through a photosystem will leave the reaction center of that photosystem oxidized. Elevating another electron will first require reduction of the reaction center. The excited electrons lost from the reaction center 700 of photosystem I are replaced by transfer from plasticyanin, whose electrons come from electron transport through photosystem Roman II. Photosystem Roman II, as the first step of the Z-scheme, requires an external source of electrons to reduce its oxidized chlorophyll reaction center. The source of electrons for photosynthesis in green plants and cyanobacteria is water. Two water molecules are oxidized by the energy of four successive charge separation reactions of photosystem Roman II to yield a molecule of diatomic oxygen and four hydrogen ions. The electrons yielded are transferred to a redox active tyrosine residue that is oxidized by the energy of 680+. This resets the ability of 680 to absorb another photon and release another photodissociated electron. The oxidation of water is catalyzed in photosystem Roman II by a redox active structure that contains 4 manganese ions and a calcium ion. This oxygen-evolving complex binds 2 water molecules and contains the 4 oxidizing equivalents that are used to drive the water oxidizing reaction Cox state diagrams. The hydrogen ions are released in the thylakoid lumen and therefore contribute to the transmembrane chemismatic potential that leads to ATP synthesis. Oxygen is a waste product of light-dependent reactions, but the majority of organisms on Earth use oxygen and its energy for cellular respiration, including photosynthetic organisms. Light-independent reactions. Calvin cycle. In the light-independent or dark reactions, the enzyme RuBisCO captures CO2 from the atmosphere and, in a process called the Calvin cycle, uses the newly formed NADP and releases 3 carbon sugars, which are later combined to form sucrose and starch. The overall equation for the light-independent reactions in green plants is 128 carbon fixation produces the 3 carbon sugar intermediate, which is then converted into the final carbohydrate products. The simple carbon sugars produced by photosynthesis are then used to form other organic compounds, such as the building material cellulose, the precursors for lipid and amino acid biosynthesis, or as a fuel in cellular respiration. The latter occurs not only in plants but also in animals when the carbon and energy from plants is passed through a food chain. The fixation or reduction of carbon dioxide is a process in which carbon dioxide combines with a 5-carbon sugar ribulose 15 bisphosphate to yield 2 molecules of a 3-carbon compound glycerate 3-phosphate, also known as 3-phosphoglycerate. Glycerate 3-phosphate, in the presence of ATP and NADP produced during the light-dependent stages, is reduced to glyceraldehyde 3-phosphate. This product is also referred to as 3-phosphoglyceraldehyde PGAL or, more generically, as triose phosphate. Most 5 out of 6 molecules of the glyceraldehyde 3-phosphate produced are used to regenerate ribulose 15 bisphosphate so the process can continue. The triose phosphates not thus recycled often condense to form hexose phosphates, which ultimately yield sucrose, starch, and cellulose. The sugars produced during carbon metabolism yield pardon skeletons that can be used for other metabolic reactions like the production of amino acids and lipids. Carbon concentrating mechanisms. On land, in hot and dry conditions, plants close their stomata to prevent water loss. Under these conditions, CO2 will decrease and oxygen gas, produced by the light reactions of photosynthesis, will increase, causing an increase of photorespiration by the oxygenase activity of ribulose 15 bisphosphate carboxylase/oxygenase and decrease in carbon fixation. Some plants have evolved mechanisms to increase the CO2 concentration in the leaves under these conditions. Plants that use the C4 carbon fixation process chemically fix carbon dioxide in the cells of the mesophyll by adding it to the free carbon molecule phosphonol pyruvate PEP, a reaction catalyzed by an enzyme called PEP carboxylase, creating the 4-carbon organic acid oxaloacetic acid. Oxaloacetic acid, or malate synthesized by this process, is then translocated to specialized bundle sheath cells where the enzyme RuBisCO and other Calvin cycle enzymes are located and where CO released by decarboxylation of the 4-carbon acids is then fixed by RuBisCO activity to the 3-carbon 3-phosphoglyceric acids. The physical separation of RuBisCO from the oxygen-generating light reactions reduces photorespiration and increases CO fixation and, thus, the photosynthetic capacity of the leaf. C4 plants can produce more sugar than C3 plants in conditions of high light and temperature. Many important crop plants are C4 plants, including maize, sorghum, sugarcane, and millet. Plants that do not use PEP carboxylase in carbon fixation are called C3 plants because the primary carboxylation reaction, catalyzed by Ru-bis-CO, produces the 3-carbon-3 phosphoglyceric acids directly in the Calvin-Benson cycle. Over 90% of plants use C3 carbon fixation compared to 3% that use C4 carbon fixation. However, the evolution of C4 in over 60 plant lineages makes it a striking example of convergent evolution. C2 photosynthesis, which involves carbon concentration by selective breakdown of photorespiratory glycine, is both an evolutionary precursor to C4 and a useful CCM in its own right. Xerophytes, such as cacti and most succulents, also use PEP carboxylase to capture carbon dioxide in a process called crassulation acid metabolism (CNM). In contrast to C4 metabolism, which spatially separates the C-ATWO fixation to PEP from the Calvin cycle, CAM temporally separates these two processes. CAM plants have a different leaf anatomy from C3 plants and fix the C-ATWO at night when their stomata are open. CAM plants store the C-ATWO mostly in the form of malic acid via carboxylation of phosphoenol pyruvate to oxaloacetate, which is then reduced to malate. Decarboxylation of malate during the day releases C-ATWO inside the leaves, thus allowing carbon fixation to 3-phosphoglycerate by Ru-bis-CO. CAM is used by 16,000 species of plants. Calcium oxalate accumulating plants such as Amaranthus hybridus and Colybanthus quitensis show a variation of photosynthesis where calcium oxalate crystals function as dynamic carbon pools, supplying carbon dioxide C-ATWO to photosynthetic cells when stomata are partially or totally closed. This process was named alarm photosynthesis. Under stress, conditions, e.g. water deficit oxalate released from calcium oxalate crystals, is converted to C-ATWO by an oxalate oxidase enzyme, and the produced C-ATWO can support the Calvin cycle reactions. Reactive hydrogen peroxide H2-ATWO, the byproduct of oxalate oxidase reaction, can be neutralized by catalase. Alarm photosynthesis represents a photosynthetic variant to be added to the well-known C4 and CAM pathways. However, alarm photosynthesis, in contrast to these pathways, operates as a biochemical pump that collects carbon from the organ interior or from the soil and not from the atmosphere. In water, cyanobacteria possess parboxysomes, which increase the concentration of C-ATWO around Ru-bis-CO to increase the rate of photosynthesis. An enzyme, carbonic anhydrase, located within the carboxysome releases C-ATWO from dissolved hydrocarbonate ions HCO-3. Before the C-ATWO diffuses out, it is quickly sponged up by Ru-bis-CO, which is concentrated within the carboxysomes. HCO-3 ions are made from C-ATWO outside the cell by another carbonic anhydrase and are actively pumped into the cell by a membrane protein. They cannot cross the membrane as they are charged, and within the cytosol they turn back into C-ATWO very slowly without the help of carbonic anhydrase. This causes the HCO- ions to accumulate within the cell from where they diffuse into the carboxysomes. Pyrenoids in algae and hornworts also act to concentrate C-ATWO around Ru-bis-CO. Order and kinetics. The overall process of photosynthesis takes place in 4 stages. Efficiency. Plants usually convert light into chemical energy with a photosynthetic efficiency of 3-6%. Absorbed light that is unconverted is dissipated primarily as heat, with a small fraction 1-2% re-emitted as chlorophyll fluorescence at longer redder wavelengths. This fact allows measurement of the light reaction of photosynthesis by using chlorophyll fluorometers. Actual plants' photosynthetic efficiency varies with the frequency of the light being converted, light intensity, temperature, and proportion of carbon dioxide in the atmosphere, and can vary from 0.1% to 8%. By comparison, solar panels convert light into electric energy at an efficiency of approximately 6–20% for mass-produced panels and above 40% in laboratory devices. Scientists are studying photosynthesis in hopes of developing plants with increased yield. The efficiency of both light and dark reactions can be measured, but the relationship between the two can be complex. For example, the ATP and NADP energy molecules created by the light reaction can be used for carbon fixation or for photorespiration in C3 plants. Electrons may also flow to other electron sinks. For this reason, it is not uncommon for authors to differentiate between work done under non-photorespiratory conditions and under photorespiratory conditions. Chlorophyll fluorescence of photosystem Roman 2 can measure the light reaction, and infrared gas analyzers can measure the dark reaction. It is also possible to investigate both at the same time using an integrated chlorophyll fluorometer and gas exchange system, or by using two separate systems together. Infrared gas analyzers and some moisture sensors are sensitive enough to measure the photosynthetic assimilation of CO2 and of delta H2O using reliable methods. CO2 is commonly measured in nmols/m2/s parts per million or volume per million, and H2O is commonly measured in mmol/m2/s or in mbars. By measuring CO2 assimilation, delta H2O, leaf temperature, barometric pressure, leaf area, and photosynthetically active radiation or PAR, it becomes possible to estimate or carbon assimilation, E or transpiration, Js or stomatal conductance, and Psi or intracellular CO2. However, it is more common to use chlorophyll fluorescence for plant stress measurement where appropriate, because the most commonly used parameters FV/FM and YII or F/FM can be measured in a few seconds, allowing the investigation of larger plant populations. Gas exchange systems that offer control of CO2 levels above and below ambient allow the common practice of measurement of A/Psi curves at different CO2 levels to characterize a plant's photosynthetic response. Integrated chlorophyll fluorometer gas exchange systems allow a more precise measure of photosynthetic response and mechanisms. While standard gas exchange photosynthesis systems can measure Psi or substomatal CO2 levels, the addition of integrated chlorophyll fluorescence measurements allows a more precise measurement of CC to replace Psi. The estimation of CO2 at the site of carboxylation in the chloroplast, or CC, becomes possible with the measurement of mesophyll conductance or GIDM using an integrated system. Photosynthesis measurement systems are not designed to directly measure the amount of light absorbed by the leaf, but analysis of chlorophyll fluorescence 700 and 515 absorbance and gas exchange measurements reveal detailed information about, e.g., the photosystem's quantum efficiency and the CO2 assimilation rates. With some instruments, even wavelength dependency of the photosynthetic efficiency can be analyzed. A phenomenon known as quantum walk increases the efficiency of the energy transport of light significantly. In the photosynthetic cell of an alga, bacterium, or plant, there are light-sensitive molecules called chromophores arranged in an antenna-shaped structure named a photocomplex. When a photon is absorbed by a chromophore, it is converted into a quasi-particle referred to as an exciton, which jumps from chromophore to chromophore towards the reaction center of the photocomplex, a collection of molecules that traps its energy in a chemical form accessible to the cell's metabolism. The exciton's wave properties enable it to cover a wider area and try out several possible paths simultaneously, allowing it to instantaneously choose the most efficient route where it will have the highest probability of arriving at its destination in the minimum possible time. Because that quantum walking takes place at temperatures far higher than quantum phenomena usually occur, it is only possible over very short distances. Obstacles in the form of destructive interference cause the particle to lose its wave properties for an instant before it regains them once again after it is freed from its locked position through a classic hop. The movement of the electron towards the photocenter is therefore covered in a series of conventional hops and quantum walks. Evolution. Early photosynthetic systems, such as those in green and purple sulfur and green and purple non-sulfur bacteria, are thought to have been anexogenic and used various other molecules than water as electron donors. Green and purple sulfur bacteria are thought to have used hydrogen and sulfur as electron donors. Green non-sulfur bacteria used various amino and other organic acids as electron donors. Purple non-sulfur bacteria used a variety of nonspecific organic molecules. The use of these molecules is consistent with the geological evidence that Earth's early atmosphere was highly reducing at that time. Fossils of what are thought to be filamentous photosynthetic organisms have been dated at 3.4 billion years old. More recent studies also suggest that photosynthesis may have begun about 3.4 billion years ago. The main source of oxygen in the Earth's atmosphere derives from oxygenic photosynthesis, and its appearance is sometimes referred to as the oxygen catastrophe. Geological evidence suggests that oxygenic photosynthesis, such as that in cyanobacteria, became important during the Paleoproterozoic era around 2 billion years ago. Modern photosynthesis in plants and most photosynthetic prokaryotes is oxygenic, using water as an electron donor, which is oxidized to molecular oxygen in the photosynthetic reaction center. Symbiosis and the origin of chloroplasts. Several groups of animals have formed symbiotic relationships with photosynthetic algae. These are most common in corals, sponges, and sea anemones. It is presumed that this is due to the particularly simple body plans and large surface areas of these animals compared to their volumes. In addition, a few marine mollusks, Elysia and Elysia chlorotica, also maintain a symbiotic relationship with chloroplasts they capture from the algae in their diet and then store in their bodies. See kleptoplasty. This allows the mollusks to survive solely by photosynthesis for several months at a time. Some of the genes from the plant's cell nucleus have even been transferred to the slubs so that the chloroplasts can be supplied with proteins that they need to survive. An even closer form of symbiosis may explain the origin of chloroplasts. Chloroplasts have many similarities with photosynthetic bacteria, including a circular chromosome, prokaryotic type ribosome, and similar proteins in the photosynthetic reaction center. The endosymbiotic theory suggests that photosynthetic bacteria were acquired by endocytosis by early eukaryotic cells to form the first plant cells. Therefore, chloroplasts may be photosynthetic bacteria that adapted to life inside plant cells. Like mitochondria, chloroplasts possess their own DNA, separate from the nuclear DNA of their plant host cells, and the genes in this chloroplast DNA resemble those found in cyanobacteria. DNA in chloroplasts codes for redox proteins such as those found in the photosynthetic reaction centers. The co-RR hypothesis proposes that this co-location of genes with their gene products is required for redox regulation of gene expression and accounts for the persistence of DNA in bioenergetic organelles. Photosynthetic eukaryotic lineages. Symbiotic and kleptoplastic organisms excluded, the glaucophytes and the red and green algae clade Archaeplastida uni and multicellular, the cryptophytes clade Cryptista unicellular, the haptophytes clade Haptista unicellular, the dinoflagellates and chromerids in the superphylum Mysozoa, and Pseudobulpharisma in the phylum Ciliophora clade Alveolata unicellular, the acrophytes clade Heteroconta uni and multicellular, the chlororetinophytes and 3 species of Polynella in the phylum Serpozoa clade Rosaria unicellular, the euglenids clade Excavata unicellular except for the euglenids, which are found within the excavata. All of these belong to the Diaphoretics. Archaeplastida and the photosynthetic Polynella got their plastids, which are surrounded by 2 membranes, through primary endosymbiosis in 2 separate events by engulfing a cyanobacterium. The plastids in all the other groups have either a red or green algal origin, and are referred to as the red lineages and the green lineages. The only known exception is the ciliate Pseudobulpharisma tenu, which in addition to its plastids that originated from green algae also has a purple sulfur bacterium as symbiont. In dinoflagellates and euglenids the plastids are surrounded by 3 membranes, and in the remaining lines by 4. A nucleomorph, remnants of the original algal nucleus located between the inner and outer membranes of the plastid, is present in the cryptophytes from a red alga and chlorarchaeophytes from a green alga. Some dinoflagellates that lost their photosynthetic ability later regained it again through new endosymbiotic events with different algae. While able to perform photosynthesis, many of these eukaryotic groups are mixotrophs and practice heterotrophy to various degrees. Cyanobacteria and the evolution of photosynthesis. The biochemical capacity to use water as the source for electrons in photosynthesis evolved once in a common ancestor of extant cyanobacteria, formerly called blue-green algae, which are the only prokaryotes performing oxygenic photosynthesis. The geological record indicates that this transforming event took place early in Earth's history, at least 2450-2,320 million years ago Ma, and it is speculated much earlier. Because the Earth's atmosphere contained almost no oxygen during the estimated development of photosynthesis, it is believed that the first photosynthetic cyanobacteria did not generate oxygen. Available evidence from geobiological studies of Archean bigger than 2,500 Ma sedimentary rocks indicates that life existed 3,500 Ma, but the question of when oxygenic photosynthesis evolved is still unanswered. A clear paleontological window on cyanobacterial evolution opened about 2000 Ma, revealing an already diverse biota of cyanobacteria. Cyanobacteria remained the principal primary producers of oxygen throughout the Proterozoic Eon 20,500–540, 3 Ma, in part because the redox structure of the oceans favored photoautotrophs capable of nitrogen fixation. Green algae joined cyanobacteria as the major primary producers of oxygen on continental shelves near the end of the Proterozoic, but only with the Mesozoic 251–60 6 Ma radiations of dinoflagellates, cockle of the phorids, and diatoms did the primary production of oxygen in marine shelf waters take modern form. Cyanobacteria remain critical to marine ecosystems as primary producers of oxygen in oceanic gyres as agents of biological nitrogen fixation, and in modified form as the plastids of marine algae. Experimental history. Discovery. Although some of the steps in photosynthesis are still not completely understood, the overall photosynthetic equation has been known since the 19th century. Jan van Helmont began the research of the process in the mid-17th century when he carefully measured the mass of the soil used by a plant and the mass of the plant as it grew. After noticing that the soil mass changed very little, he hypothesized that the mass of the growing plant must come from the water, the only substance he added to the potted plant. His hypothesis was partially accurate, much of the gained mass comes from carbon dioxide as well as water. However, this was a signaling point to the idea that the bulk of a plant's biomass comes from the inputs of photosynthesis, not the soil itself. Joseph Priestley, a chemist and minister, discovered that when he isolated a volume of air under an inverted jar and burned a candle in it which gave off CO2, the candle would burn out very quickly, much before it ran out of wax. He further discovered that a mouse could similarly injure air. He then showed that the air that had been injured by the candle and the mouse could be restored by a plant. In 1779, Jan Ingenhouse repeated Priestley's experiments. He discovered that it was the influence of sunlight on the plant that could cause it to revive a mouse in a matter of hours. In 1796, Jean Senebier, a Swiss pastor, botanist, and naturalist, demonstrated that green plants consume carbon dioxide and release oxygen under the influence of light. Soon afterward, Nicolas Theodora de Saussure showed that the increase in mass of the plant as it grows could not be due only to uptake of CO2, but also to the incorporation of water. Thus, the basic reaction by which photosynthesis is used to produce food such as glucose was outlined. Refinements. Cornelis van Niel made key discoveries explaining the chemistry of photosynthesis. By studying purple sulfur bacteria and green bacteria, he was the first to demonstrate that photosynthesis is a light-dependent redox reaction in which hydrogen donates its atoms as electrons and protons to carbon dioxide. Robert Emerson discovered two light reactions by testing plant productivity using different wavelengths of light. With the red alone, the light reactions were suppressed. When blue and red were combined, the output was much more substantial. Thus, there were two photosystems, one absorbing up to 600 nm wavelengths, the other up to 700 nm. The former is known as SEI, the latter is PSI. PSI contains only chlorophyll, SEI contains primarily chlorophyll with most of the available chlorophyll B, among other pigments. These include phycobilins, which are the red and blue pigments of red and blue algae, respectively, and fucoxanthol for brown algae and diatoms. The process is most productive when the absorption of quantities equal in both SEI and PSI, assuring that input energy from the antenna complex is divided between the PSI and SEI systems, which in turn powers the photochemistry. Robert Hill thought that a complex of reactions consisted of an intermediate to cytochrome B6, now a plastoquinone, and that another was from cytochrome F to a step in the carbohydrate generating mechanisms. These are linked by plastoquinone, which does require energy to reduce cytochrome F. Further experiments to prove that the oxygen developed during the photosynthesis of green plants came from water were performed by Hill in 1937 and 1939. He showed that isolated chloroplasts give off oxygen in the presence of unnatural reducing agents like iron oxalate, ferrocyanide, or benzoquinone after exposure to light. In the Hill reaction, 2H2O plus to a plus light, chloroplasts 2H2 plus OTWO is the electron acceptor. Therefore, in light, the electron acceptor is reduced and oxygen is evolved. Samuel Rubin and Martin Kamen used radioactive isotopes to determine that the oxygen liberated in photosynthesis came from the water. Melvin Calvin and Andrew Benson, along with James Basham, elucidated the path of carbon assimilation, the photosynthetic carbon reduction cycle in plants. The carbon reduction cycle is known as the Calvin cycle, but many scientists refer to it as the Calvin-Benson, Benson-Calvin, or even Calvin-Benson-Basham or CBB cycle. Nobel Prize-winning scientist Rudolf Marcus was later able to discover the function and significance of the electron transport chain. Ito Heinrich Warburg and Dean Burke discovered the I-quantum photosynthesis reaction, that splits C-Otwo, activated by the respiration. In 1950, first experimental evidence for the existence of photophosphorylation in vivo was presented by Ito Candler using intact chlorella cells and interpreting his findings as light-dependent ATP formation. In 1954, Daniel I. Arnon et al. Discovered photophosphorylation in vitro in isolated chloroplasts with the help of p32. Louis N.M. Dysons and Jan Amis discovered that chlorophyll will absorb one light, oxidized cytochrome F, while chlorophyll and other pigments will absorb another light but will reduce this same oxidized cytochrome, stating that two light reactions are in series. Development of the concept. In 1893, Charles Reid Barnes proposed two terms, photosyntax and photosynthesis, for the biological process of synthesis of complex carbon compounds out of carbonic acid in the presence of chlorophyll under the influence of light. Over time, the term photosynthesis came into common usage. Later discovery of annexogenic photosynthetic bacteria and photophosphorylation necessitated redefinition of the term. C3-C4 photosynthesis research. In the late 1940s at the University of California, Berkeley, the details of photosynthetic carbon metabolism were sorted out by the chemists Melvin Calvin, Andrew Benson, James Basham, and a score of students and researchers utilizing the carbon-14 isotope and paper chromatography techniques. The pathway of C-Otwo fixation by the albichlorella in a fraction of a second in light resulted in a 3-carbon molecule called phosphoglyceric acid PGA. For that original and groundbreaking work, a Nobel Prize in Chemistry was awarded to Melvin Calvin in 1961. In parallel, plant physiologists studied leaf gas exchanges using the new method of infrared gas analysis and a leaf chamber where the net photosynthetic rates ranged from 10 to 13 μmol C-Otwo M-2 mi, with the conclusion that all terrestrial plants have the same photosynthetic capacities that are light saturated at less than 50% of sunlight. Later, in 1958-1963 at Cornell University, field-grown maize was reported to have much greater leaf photosynthetic rates of 40 μmol C-Otwo M-2s-1 and not be saturated at near full sunlight. This higher rate in maize was almost double of those observed in other species such as wheat and soybean, indicating that large differences in photosynthesis exist among higher plants. At the University of Arizona, detailed gas exchange research on more than 15 species of monocot and dicot uncovered for the first time that differences in leaf anatomy are crucial factors in differentiating photosynthetic capacities among species. In tropical grasses, including maize, sorghum, sugarcane, Bermuda grass, and in the dicot amaranthus, leaf photosynthetic rates were around 38-40 μmol C-Otwo M-2s-1, and the leaves have two types of green cells, i.e., outer layer of mesophyll cells surrounding a tightly packed chlorophyllous vascular bundle, sheath cells. This type of anatomy was termed Krantz anatomy in the 19th century by the botanist Gottlob Haberlandt while studying leaf anatomy of sugarcane. Plant species with the greatest photosynthetic rates and Krantz anatomy showed no apparent photorespiration, very low C-Otwo compensation point, high optimum temperature, high stomatal resistances, and lower mesophyll resistances for gas diffusion and rates never saturated at full sunlight. The research at Arizona was designated a cetacean classic in 1986. These species were later termed C4 plants, as the first stable compound of C-Otwo fixation in light has 4 carbons as malate and aspartate. Other species that lack Krantz anatomy were termed C3 types such as cotton and sunflower, as the first stable carbon compound is the 3-carbon PGA. At 1000 ppm C-Otwo in measuring air, Both the C3 and C4 plants had similar leaf photosynthetic rates around 60 μmol, C Otwell M-2S-1, indicating the suppression of photorespiration in C3 plants. Factors. There are three main factors affecting photosynthesis and several corollary factors. The three main are light irradiance and wavelength, carbon dioxide concentration, temperature. Total photosynthesis is limited by a range of environmental factors. These include the amount of light available, the amount of leaf area a plant has to capture light. Shading by other plants is a major limitation of photosynthesis, the rate at which carbon dioxide can be supplied to the chloroplasts to support photosynthesis, the availability of water, and the availability of suitable temperatures for carrying out photosynthesis. Light intensity, irradiance, wavelength, and temperature. The process of photosynthesis provides the main input of free energy into the biosphere and is one of four main ways in which radiation is important for plant life. The radiation climate within plant communities is extremely variable in both time and space. In the early 20th century, Frederick Blackman and Gabrielle Matthey investigated the effects of light intensity, irradiance, and temperature on the rate of carbon assimilation. At constant temperature, the rate of carbon assimilation varies with irradiance, increasing as the irradiance increases, but reaching a plateau at higher irradiance. At low irradiance, increasing the temperature has little influence on the rate of carbon assimilation. At constant high irradiance, the rate of carbon assimilation increases as the temperature is increased. These two experiments illustrate several important points. First, it is known that, in general, photochemical reactions are not affected by temperature. However, these experiments clearly show that temperature affects the rate of carbon assimilation, so there must be two sets of reactions in the full process of carbon assimilation. These are the light-dependent photochemical temperature-independent stage and the light-independent temperature-dependent stage. Second, Blackman's experiments illustrate the concept of limiting factors. Another limiting factor is the wavelength of light. Cyanobacteria, which reside several meters underwater, cannot receive the correct wavelengths required to cause photoinduced charge separation in conventional photosynthetic pigments. To combat this problem, a series of proteins with different pigments surround the reaction center. This unit is called a phycobilizum. Carbon dioxide levels and photorespiration. As carbon dioxide concentrations rise, the rate at which sugars are made by the light-independent reactions increases until limited by other factors. Rubis-Co, the enzyme that captures carbon dioxide in the light-independent reactions, has a binding affinity for both carbon dioxide and oxygen. When the concentration of carbon dioxide is high, Rubis-Co will fix carbon dioxide. However, if the carbon dioxide concentration is low, Rubis-Co will bind oxygen instead of carbon dioxide. This process, called photorespiration, uses energy, but does not produce sugars. Rubis-Co oxygenase activity is disadvantageous to plants for several reasons. One product of oxygenase activity is phosphoglycolate 2-carbon instead of 3-phosphoglycerate 3-carbon. Phosphoglycolate cannot be metabolized by the Calvin-Benson cycle and represents carbon lost from the cycle. A high oxygenase activity, therefore, drains the sugars that are required to recycle ribulose 5-bisphosphate and for the continuation of the Calvin-Benson cycle. Phosphoglycolate is quickly metabolized to glycolate that is toxic to a plant at a high concentration. It inhibits photosynthesis. Salvaging glycolate is an energetically expensive process that uses the glycolate pathway, and only 75% of the carbon is returned to the Calvin-Benson cycle as 3-phosphoglycerate. The reactions also produce ammonia NH3, which is able to diffuse out of the plant, leading to a loss of nitrogen. A highly simplified summary is 2-glycolate plus ATP3 phosphoglycerate plus carbon dioxide plus ADP plus NH3. The salvaging pathway for the products of rubus-CO oxygenase activity is more commonly known as photoRET, since it is characterized by light-dependent oxygen consumption and the release of carbon dioxide.
Thank you for watching. Consider supporting the channel, leave a like and subscribe.